All right, thank you very much. Title is Interpretation of Compositional Data for Feed. And I've spent some 30 years in FDA working on the subject and working with the OECD. So this presentation is based on what has been observed from the note to files posted on the FDA website under completed consultations. Also, it's based on my experience as a FDA regulator and working with different people. Made some notes this morning I thought might be interesting to go with those. First, when we start talking about policy, laws, regulations, guidance, we have to understand that there is some world guidance through the Codex Alimentarius. While it says it's not applicable to feeds, it really is. It is there. It has been through WTO, WHO, it's recognized. So countries that are regulating are, in order to be recognized through the WTO, have to comply with what those guidance is, at least in stature. So if anybody's going to change anything, that's a body one seems like they would have to talk to make some differences there. The OECD certainly is big for the white papers, like the task force with plant composition and all. And that is a reasonable body to work with. One thing one has to remember about that is they're represented, there's, I don't know how many countries are on all, there was 27, it's probably 30 now. But these are individual countries, not the EU per se, but representing their own countries. So while, and they seem to be quite amiable and agreeing with what's in the OECD papers and all, it doesn't mean that their regulatory bodies are going to follow that, because they have to follow what the EU is following. In the US, the policy in 92, guidelines in 96, that hasn't been changed, so that remains pretty well constant. In Canada, Lynn Underhill and I, in writing our paper from the Composition Task Force, 93 for novel foods, 97 or 95, 08, and revised in 12 with the CFIA. So those things are in place, all right? So to change things from that standpoint, these are the types of things one needs to look at. You just don't say that you're going to do it on a review basis, because they have to back up to what they've published and what are in the guidance. There's talk on the variability, you know, variation is ubiquitous. It's everywhere, everybody knows that. They knew it when they developed statistics. Statistics was a way to deal with variation. And my understanding from my training was that statistics for agricultural products anyway started with barley, all right? And selecting barley for a certain amount of sugars for the beer making industry. So they wanted to make sure that when they bought barley, that they knew how much sugar were in there to be able to make the barley. So they developed statistics to deal with the variation, all right? Now, a colleague from the UK in our composition workshop, David Lovell, was trying to point us away from comparative statistics to predictable statistics. And rather than doing this point-to-point comparisons, more predictions with confidence intervals and things like that. So that's dealing more with variability, which we know is there. And when we look at a group of variability, then you look and see the probability of what's happening with availability and make a prediction based on the variability that's there, what the outcomes are going to be. So that's kind of what as far as my notes from this morning, is things to think about. So the crop composition database is one database that's using those predictability things. So the focus of my talk now, getting away from regulations and all that, is on composition. So we heard this morning from Kevin and others that you got intended effects and unintended effects that we're looking at composition. So that's pretty well been covered. So the unintended effects was a way of looking at things that we couldn't predict that were going to happen. That's solely seemed to be what we were doing that for. And I look at this, and when in FDA, when we were getting together in '92, policy, the statement of policy, we had David Kessler, Mike Taylor, Jim Mariansky, Eric Flam, a lady by the name of Linda Call, a lawyer by the name of Karen Kopp, that we tossed all these things around and were thinking, well, this thing should be sunsetted in 5 years. So if you look back to the OECD and looking at what key nutrients, everybody understands what's been published there. The U.S. National Research Council has scientific reports on nutrient requirements of domestic animals, since we're talking about feed. There's also one for humans on nutrient requirements. Anyhow, this kind of serves as a key of deriving what key nutrients, anti-nutrients, natural toxicants, and so forth are in animals, and humans as well. Key nutrients are defined as proximates, amino acids, fatty acids, calcium, and phosphorus. Key anti-nutrients defined by OECDs as adversely affect metabolism, such as trypsin inhibitors in soybeans. They bind minerals, make them unavailable to the animal, like phytic acid in corn and other crops. And they cause adverse effects to the animals, like lectins in soybeans. Key toxicants, just as a point in case, would be gospel in cottonseed and silogenic glycoside in cassava. We heard from Wilna Van Leucine from South Africa gave a paper, and when they come back to write their manuscript, they were saying that cassava was not a recognized safe crop for people to eat. We said, well, there's like 10 billion people eating that, you know. So they finally backed off that. And so you have to recognize under whatever the processing things are to make the crop safe, that's what we consider as the safe, or at least what people are consuming. What are proximates? Well, moisture is just something you go to take off, and then you go look at your crude protein. And I mentioned the figure this morning when we were talking about the nitrogen times 6.25, and animal nutrition are pretty well use that, but if you get more specific, because that was just an estimated amount of real protein versus amount of nitrogen in the plant. So there are different types of nitrogenous products other than proteins and amino acids in the plant that are not in the protein category. We got crude fat is usually ether extractable. Ash is what remains after combustion in a muffle furnace at 500 degrees Celsius. And fiber is remaining after acid treatment and drying. So crude fiber is something that's been used many times in the past, and it's an obsolete measurement, yet we have like pet food requires it and all that, and it just seems to be a standard measurement. It's not really in nutrition, it's not really used anymore. Your nutritional books will say that. We got total dietary fiber now is what's used in humans nutrition. We have acid detergent fiber and neutral detergent fiber. So these are what are very important in the animal nutrition. Amino acids, we got essentially, if we talk about essential amino acids, we got what we call a HIL, MP, TVT, right? So anybody that learned their early nutrition would learn that's acronym, so to speak, and so they're usually considered essential for non-ruminant animals, and then we got glycine, tyrosine, and serine are also important. Some people think glutamic acid or glutamine is also sometimes. We have the dietary supplement people make you think that everything is important, but I think in writing the OECD papers we kind of come down to this part. What are the fatty acids? So some fatty acids are essential in the diet for the animals, like linoleic, linolenic. Others of interest is gamma-linolenic, which is the one of interest for humans now, and the palmitic and palmitoleic. So omega-3s is what you're going to hear a lot about in the future, and that's the gamma. Okay. Major minerals include calcium, phosphorus, magnesium, potassium, and sodium. Trace minerals include iron, selenium, manganese, copper, and zinc. Other than calcium and phosphorus, the key minerals depend on what the feed from the unmodified plant is recognized for. Most of the time in animal nutrition, it's calcium and phosphorus that are of real interest, because most of the other ones somewhat dependent on the environment, where they're grown as far as the level and whether or not it's going to meet the animal's requirements. And particularly selenium is a peculiar element, because if we start in on the west coast and come to the east, the more acid, the more the selenium gets leached out. And where prior to irrigation, selenium was pretty high on a lot of soils in the west, and there were selenium holes where poisoning of cattle and others resulted. But in dealing with that, as the irrigation increases, it leaches more of the available selenium out. So selenium actually needs to be supplemented in many of the animal diets. And there's people who point out that humans are lacking selenium too and need to be supplemented. The vitamins A, D, E, and K is fat soluble, and the water soluble is B1, B2, B6, C, folacin, niacin, pantothenic acid. Key vitamins depend on what the feed from the unmodified plant is recognized for. You note that B12 is not produced by plants. Okay, experimental design, composition of the studies, and this is from what usually has happened in looking at your BNS or biotech notification files that are in the FDA Center for Food Safety database. And a lot of you have been doing this, so you already know it. I don't need to tell you anymore, but just a quick review. The location is generally selected to represent a geographical area where the plants expect to be grown. You go to grow corn in the Midwest, is a major place where it's going to be grown. be the irrigated parts of Nebraska, Kansas, off into the west of Arizona and California. So there's usually 3 or more blocks containing 2 or more plots are formed to ensure that the treated and controlled plants are grown under the same conditions. Cautions have been taken to ensure no cross-fertilization or other outside conditions influence the outcome. In other words, you want just as few differences between those as you can get. The more difference you get, the bigger the variation, certainly the less likely you are to show any differences. So, harvest, storage, processing is ordinarily conducted in such a manner to maintain the integrity of the experimental plant material. So, taking care of it, getting samples, taking care of those samples, get them under refrigeration so they don't deteriorate while they're being held before processing. And then once you're processing them so not to upset the integrity of the ingredients you're looking at. Composition analysis is usually conducted using validated analytical methods, and that's one thing that our ILSI database and Ray Shilto back there is certainly interested in making sure it happens. Because you have to list the methods have to be validated, they have to be validated in the lab that's doing the analysis so that we make sure that we're getting a valid method. One of the problems with some of the older databases, we never knew what methods they were using. We didn't know whether the labs were doing the procedures correctly or if they had validated their lab with that procedure. So the values coming out were sometimes suspect. The resulting data is mostly subjected to an appropriate statistical analysis resulting probability estimates, and we talked about that. There are certainly different statistical methods one can use to arrive at the same point. If we go back to the t-test, which where Barley started using t-test, that's the simplest thing. But we get into the analysis of variance and then other predictabilities establishing confidence limits. This is one thing, this is probably not for you all, but it's for the public, is to let them know in the US and in Canada that sponsors submit data and furnish safety assessment of new plant variety, and they have a file that is scientific, and so that's called a biotech notification file for US. FDA evaluates for all known and suspected hazards and potential intended effects on plant composition and nutritional properties. And what in the past we've told them, I'm still sure they're asking, no hanging chads. If you raise a question, you have to answer it, all right? Otherwise, it's not complete. And you all know that our system in the U.S. is supposedly voluntary, but I would question that it really is, since I think our legal system and your clients whom you're selling seeds to and the people buying the food from those people they're growing the seeds have to satisfy the buyers, and the buyers are going to demand that you have an FDA letter. And you have to have your EPA clearance, John Cowell says there, and you have to have your USDA exceptions. So, data is usually presented from assembled in a table form, and they have averages, P's and F, and I didn't put on there that a lot of times would be the confidence limits. And we're talking about being consistent with a recognized database. They're usually given a % of dry weight to have a standard form of presentation. With the exception of fatty acids, they're usually given a % of total fatty acids. And amino acids can also given on a percent of, are also given on a percent of total protein, but since you know the protein, then one can figure back the actual amount of amino acid in the crop, in the seed. And this is generally what has been done from a feed standpoint. The expressed protein is not a toxin or related to a known toxin, and other components are not adversely affected. There's no compounds produced or expressed that would require a food additive petition because of being unsafe. And you have to remember in the US, that's the deciding factor of the food additive petition from an FDA standpoint. If there's nothing in there requiring that, then there's no preclearance required. Key nutrients, anti-nutrients, and actual toxicants normally present in the bioengineered plant are not adversely changed. And underline key because that's important, because not all of those are key. All right, so that's the reason to go back to the OECD on what key is all about. So the experimental comparator, all right, then we heard Phil talked about that this morning and others talked about it, so I won't talk any more about that. And this is generally, and Phil was talking about this, if there's a significant difference found, go to the recognized database. So we got one database that wasn't mentioned was USDA is a standard reference database, and that one is one that is pretty complete, and it's well financed through, it was anyway, unless it was sequestered. So they do keep that database up to date. Okay. So in the bottom line is the logical scientific justification has been presented involving safety data or new safety data not previously available. So once you get through all that, there's usually nothing left. I mean, everything passes when we get through those. Yeah. All right. Weight of evidence, all right. So that means just you have to go through, if you do find some differences, and explain what's happened, all right? All right, this will go through rather quickly. This is what I picked off the BNF file. So we got alfalfa only has 1, bent grass 1, canola's got 11. Transformation events, there may be a couple or 3 transformation events within each of the biotech notification files. Corn has gotten the most of any. Cotton has got quite a few, and flax. Number of sponsors kind of changes based on who buys who out, and some of the earlier sponsors are bought out by others, so they're probably not pertinent to talk about those. Soybean, and it's got that. And you got cantaloupe, pitaya, and these are not usually used for feed. So we got a total of what I counted up, 93 and 150 transformation events. If we look at the traits, we got herbicide, pest resistant in both. So we got, look at that. We got with what's happening with those. And so the conclusion is over the last 20 years, completed 193 consultations, 150 transformations, and 11 crops. And so we have not identified any FDA has not identified any safety regulation issues under the FD&C Act that require further evaluation. References, we got the cystine database. Rod Herman and I wrote a paper. Rod did most of the work. And then Lynn Underhill and I have a paper we're trying to get published from the workshop. All righty.